Hi viewers, my class is today module 1 is introduction to proteomics. So now I am going to tell you the brief introduction about the proteomics and their importance. So the term proteome and the proteomics were coined by the Mark Wilkins and colleagues in the early 19s. The aim of the proteomics to identify all the proteins in a cell to create a complete three-dimensional 3D map of the cell indicating where the proteins are located. So proteomic is also known as a omics. The main omics nomenclature is genomics. So genomics means study of the DNA. So next again DNA in transcription it converted to RNA. Study of RNA is called transcriptomic. Again RNA synthesized as a protein, that is called the translation. So synthesis of the protein is the study of proteomics. By combination of the transcriptomics and proteomics is the study of the functional genomics. So proteins act as a functional component in enzymatic reactions, that is called the metabolites. So study of the metabolites is called metabolomics. So proteomics is the study of overall state of an organism's temporal protein composition. The biological state of the proteome is encoded in the relative abundance of currently expressed protein and their isoform, their localization related to cellular, structural, their interaction pattern, molecules and substrate, their current post-translational modification state, their folded structural form. Proteomes are all the dynamic structures in the metabolism. So what are the steps involved in the gene expression control? There are the several steps including in the nucleus and cytosol. And coming to the nucleus it converts the DNA to primary RNA transcript, RNA to mRNA. So conversion of DNA to primary RNA transcript is called the transcriptional control, and primary transcript to the mRNA is called the RNA processing control. So once the mRNA has been formed, it transported into the cytosol. In the cytosol, RNA undergo the different pathways. mRNA once it reaches to the cytosol, it convert into two different forms, in inactive mRNA form and the protein form. So from mRNA to inactive mRNA form is called RNA degradation control mechanism. mRNA to protein formation is under the translational control mechanism. Protein to modified protein is the post-translational modification. So overall the steps involved in this gene expression control, these are the main steps involved in the gene expression control mechanism. So there is a different types of the proteomics. Now coming to the types of the proteomics. The current proteome investigations are essential, mainly focused on the two major areas. The first one is expression proteomics, which aim to measure the upregulation and downregulation of the protein level. Functional proteomics, which aims to characterize the protein activities, multi-protein complexes and signaling pathways. Okay, so next protein expression proteomics. So what is the main aim of the proteomics during the protein expression? So proteomics mainly involved in the quantitative study of the protein expression, which occurs between the samples that differ by the some variable is known as the expression proteomics. So typically expression proteomics are mainly involved in the studies. They are addressed to the investigation of the expression protein pattern in abnormal cell, that is malignant cell. That malignant cell is stimulated by the drug treatment, etc., and also in comparison with the normal cell. So in biomedical application, these proteomics are involved in the study of the comparative approach is usually employed to identify the proteins that are upregulated or downregulated in a disease specific manner for use as the diagnostic markers or therapeutic targets. So another main functioning of the proteomic study is the functional proteomics. Functional proteomics means how the proteomic study is mainly focused on the functional environment. It is mainly focused to monitor and analyze the spatial and temporal properties of the molecular network and fluxes involved in the living cells. Functional proteomics approaches are addressed towards two major targets. One is elucidation of the biological functions of unknown proteins and the definition of the cellular mechanism at the molecular level. So functional proteomics, another important is in the cell, many proteins display their biological functions through the rapid and transient association within large protein complexes. So one is understanding protein function as well as unraveling molecular mechanism within the cell, then depends on identification of interacting the protein partners. And another important thing is the association of unknown protein with a partner belong to a specific protein complex. Those are involved in the particular mechanism that would in fact be strongly suggestive of its biological function. Furthermore, a detailed description of the cell or signaling pathway might greatly benefit from the elucidation of the protein-protein interaction in vivo condition. So this is the general brief introduction about the proteomics. In next class, I will give the discussion about the what is the main introduction and functions of the proteomics. So thank